Okay, let me trail out on that integration. Couple of little subtle points on integration, then we'll move to something else, is synaptic potentiation. Synaptic potentiation basically has to do with the presynaptic, okay? Repeated use of increased efficiency, that means keep firing this presynaptic. The calcium concentration, remember we talked about, go back to electrophys, comes in to cause the release of neurotransmitter. Remember at the presynaptic, calcium comes in, that's what causes this, okay? So if you're doing a lot, firing pretty quickly, you're getting a nice accumulation of calcium in here. Get a nice accumulation because it's the same thing. The EPSPs are long in duration and action potentials. So that potentiates further release of neurotransmitter because you're building up so much calcium. Calcium concentration increases in presynaptic terminal and postsynaptic neuron, and that causes this to happen there. Okay? We'll go a little further. Integration, presynaptic inhibition. Okay. Assume that you had a neuron terminating on this axon, like you see here, an axo-axonic. Then this neuron, which is trying to release EPSP stuff, that neuron right there, trying to do it, this straight neuron, could be inhibited because if you keep firing an inhibitory on right here on this axo-axonic, you would lessen the amount of neurotransmitter for EPSP being fired. So release of excitatory by one may be inhibited by the activity of another. So here would be the EPSP, but this one is doing an IPSP directly on the axon. Less neurotransmitter would be released by this neuron right here because you're getting some inhibition from it right there. Okay? All right, let's go further now. Now we're going to talk on neurotransmitters. There are many, 50 or more, it's close to 100 have been identified, neurotransmitters. This is an old one. And they are released, you know, it used to be an old thing called Dale's principle where only one neuron could release one neurotransmitter. That's not really true anymore. We find that they can release different kinds of neurotransmitters. It's not just one and so forth. Okay. Acetylcholine, that was the one that's in the autonomic nervous system we're going to talk about, and also were the ones that went to skeletal muscle. It's synthesized in the cell body from acetate plus choline, and acetyltransferase is the enzyme. So in the neuron cell body, if this was a somatic motor neuron, in the neuron cell body, you would synthesize acetylcholine from acetate and choline. The enzyme that would hook them together is acetyltransferase. Then you transport the stuff down to the terminal. The enzyme that breaks it down is acetylcholinesterase. Remember, three things terminate neurotransmitter: selective reuptake, wash away at the cleft, or enzymatic breakdown. So acetylcholinesterase is the one that breaks it down. That's an important aspect because on certain conditions, we inhibit acetylcholinesterase in medicine, like with a condition called myasthenia gravis. So this is important to know for your clinical later. Okay. The biogenic amines, all of the amines come from tyrosine. Tyrosine, particularly these amines here, the catecholamines, come from tyrosine. Okay, class, we're back. What we're looking at now is the biogenic amines. Amines means made from one amino acid. The biogenic amines made from one amino acid include the catecholamines and the endolamines. The catecholamines and the endolamines. All right. Let's look here. All right. The endolamines, like serotonin, is going to come from tryptophan. Serotonin, because it has an endo ring. It has an endo ring. So serotonin is called 5-hydroxytryptophan. 5-hydroxytryptophan is serotonin. Now, Then histamine comes from something called histidine, but it also has this endo ring right here. The catecholamines come from tyrosine. Here's tyrosine. This right here is called the catechol ring. The catechol ring. So the catecholamines, the amines, comes from tyrosine. And the endo ring comes from tryptophan. Okay. All right, right there. All right. Now, let me just show you on the catecholamines. Okay. So here's tyrosine. And see, we did this in the skin chapter. I was showing how melanin. So tyrosine, which is an amino acid. Remember, an amino acid can be used for bioconversion or to make proteins or as a last resort for energy. You never want to break down your proteins for energy. But see, enzymes govern the direction of all biochemical reactions. We've hit this many times. So here would be tyrosine, instead of making melanin, making dopamine. And then another enzyme would come in and allow it to make norepinephrine. Then another enzyme would come in and allow it to make epinephrine. Okay, just showing you all of this. But all of these have the catechol ring. That's why they call them in clinical the catecholamines. Okay? Play roles in emotional behavior. See, like for example, for depression, sometimes we can give you SSRI, selective serotonin reuptake inhibitor. You may have heard of this. Okay? So various things of that nature there. Okay? A tricyclic antidepressant, or monoamine oxidase inhibitor. Let's move on here. Then we have some amino acids that basically are just amino acids, but they yet are acting as what? So here's glycine. These are acting as neurotransmitters. Glycine, so forth like that you can see there. Okay. So again, it's just various ones. Here's glutamate. Aspartate here. Okay. So there are many different kinds we found. Peptides include substance P for pain, mediator of pain signals. Endorphins, these are your natural high substances. The endorphins and the enkephalins. They're opiate based, but you make them yourself. So you need some natural high substances. Think about it. Just moving around and your heart beating and your joints moving, you wouldn't be able to think about nothing that would hurt. But those endorphins squelch it out. That's why overdose. See, whatever the body makes and you try to take externally, the body backs off. So the more a person takes morphine or other things like that, some opiates, then the body makes less. So if they try to come cold turkey, it is horrible, because now they aren't hardly making any themselves. Somatostatin, cholecystokinin, these are other peptides, and they do other things besides just neurotransmitter. Somatostatin has to do with growth hormone in addition. Cholecystokinin is the one that causes the gallbladder to contract. But it also has other functions. Okay, let's go further. Purines, such as ATP. Think about it. See, a chemical can be used several ways. Okay? This is for energy, but it can also be used as a neurotransmitter. Everything depends on what the receptor receives. And here's the situation. The receptor. If a neuron has an ATP receptor, then it will use it as a transmitter. Get nitric oxide. That's a new one. Guy won the Nobel Prize for this. Nitric oxide. Okay. So we find a lot of stuff here. Cannabinoids. These are the ones. This is the one that you may endocannabinoid. This is the one with the marijuana thing. Okay. See, you get whatever you take and it alters the brain or alters this, it had to have receptors in the brain. So the reason that marijuana would work is because we do make some stuff ourselves, our body, that is working off of those receptors. Okay. Then we have some that are more EPSP versus IPSP type. GABA and glycine are usually IPSPers. Acetate and glutamate are generally, which I don't see here, are generally EPSPs. Here's glutamate and then aspartate too. Acetylcholine, you know that's the one that turned on the skeletal muscle. But it inhibits cardiac muscle. But see, skeletal muscle is the somatic. Cardiac muscle is working off the autonomic. We're going to get to that later. So, but you got to have receptors okay. Some neurotransmitters work direct. Okay. And some work by what we call secondary messengers. Now, I mentioned that a little bit in the cell chapter. We won't get heavy into that. I'll do more of that in ANP2 when we need it. But some ligands work, ligand-gated, work direct. In other words, the neurotransmitter sits there and opens up the gate directly, or closes the gate directly. The indirect means it gets in there, but it has to turn on a secondary messenger to do the job. It has to go internal. Okay? So we have the ligand-gated channels, but some work by having to use this. See, this was a ligand-gated, and it opens directly. But then we have the kind that have to use a secondary messenger, like a G protein or a cyclic AMP to do it. So some work direct, some work indirect. The ones that work indirect take longer to act, but they can turn on more on the secondary messenger aspect. So the key for you is just to know some work direct and some work indirect. And then you can read on it also. Okay, let's go further. Okay, so that's just turning on a secondary messenger. Okay, so where we are now is to neuronal pools, but let's just review real quick. So we've got several different kinds of neurotransmitter substances, right? Several different kinds. We now know, unlike it used to be in the old days with Dale's principle, that a neuron can make several. In order for a neurotransmitter to work, that whatever it's going to must have receptors. Whatever it's going to must have receptors. They're different kinds. They're different kinds. Now, one way of also saying it, we have those that are more lipid soluble and water soluble. The more water soluble generally have to use a secondary messenger if they need to get to the nucleus. But anyway, we have different kinds. Acetylcholine, biogenic amines we talked about. They come from what amino acid? Okay. Different amino acids themselves can actually work direct. Different peptides. Remember, peptide is a short protein type of thing. Then we got endorphins. Okay. We got the leptins. You see, you don't see that. The leptins are the ones that make you have satiety, where you're not hungry anymore. So we're only naming a few here. Purines, such as ATP. Okay. Work. But purines, gases, nitric oxide, so forth. So we have many different ones, and rather than just learning them, ad infinitum, it's best to learn them when we need to use them for different situations, either in ANP1 or ANP2. Just like you learned acetylcholine with muscle. Okay, you could relate then, rather than just learning a list, which doesn't really, it helps and it doesn't help. Okay, direct, some work direct. Example, acetylcholine and amino acids. And then some work indirect. Okay, biogenic amines, neuropeptides, they have to turn on that secondary messenger. So that just kind of gives you an idea of how that works. And so now we're going to go to neuronal pools in another video. Thank you.